I think I just realized I did the entire intro on mute.
Oh yeah, I could not hear anything.
That's great.
Was
Well, I had a good dry run on the intro then. All right, everybody. It's 9:03. I wondered why nobody seemed to be reacting at all. So uh, it's 9:03 past the start time. So sorry for a little bit late, but that's because I was speaking into the microphone on mute. So it's my great pleasure to introduce Dr. Letizia Marvaldi, who's going to be giving our talk today. Uh, she did her PhD at Innsbruck University in Austria, and then moved to the Weizmann Institute of Science, where she's been a postdoc in Mike Fainzilber's lab. And I see Mike is also on. So hi, Mike. And she's done some really great work there. One of her first papers that I saw from her postdoctoral work was one of my favorite papers of the last several years, which was a paper published in Science about 2 years ago, where she was involved in a study demonstrating that axonal injury leads to rapid translation of mTOR in axons and how this plays a role in regeneration response. Really cool paper with, I think, some really good implications for pain. And now she's been involved in a study that's going to come out in a couple of weeks in a journal that we've all heard of about importins, which are nuclear import proteins and the role that they play in chronic pain. So it's really my pleasure to introduce Dr. Marvaldi, and please take it away. Oh, and one more thing on questions. I'm sure there'll be lots of questions, but I think maybe because of the online format, we can hold those till the end. So please write your questions down, and then we'll have plenty of time for discussion at the end. All right, Letizia.
Thank you, Ted. So, hello everybody, and good morning to you. Thank you, Ted, for the kind description of me, of my work, and for the invitation to give our talk today. I'm really glad and honored to speak about my postdoc work in Mike's lab. So in these 6 years, I worked to study the role of importin alpha-3 and the regulation of importin alpha-3 in chronic pain. And I would like to show you a few slides of introduction and present you the importins, which are the major protein that we investigated in the lab of Mike's. So the importins are nucleocytoplasmic protein. As you can see here, we are studying importin alphas and importin beta. So importin alpha in red, importin beta in blue, they form an heterodimero in the cytoplasm, and then importin alpha is binding to an NLS sequence of a protein cargo, and together this complex goes to the nucleus through the pore, to the nuclear pore, and then release the transcription factor and for the activation of genes. So gene activation, and then for the function of a cell. So, moreover, importin beta mRNA is locally translated upon injury in the syactin nerve and is forming a complex with importin alpha. So importin beta and importin alpha forming a complex together with a motoprotein called dynein, and the importin alpha is also binding to the NLS sequence cargo protein. Altogether, this complex is called retrograde transport complex that is going back to the nucleus for the activation of genes and then the neuronal function. Also in the lab, we are interested in the neuronal regeneration and local translation, as well as cell size regulation. Another topic we are interested in are the cargo specificity of the importin alpha. So there are 6 importin alpha in the mouse model, and each importin alpha can bind to a specific cargo, a transcription factor, that then is going to activate in the gene expression for the function of a neuron. And since we are interested specifically in sensory neurons, in our work we started to work with the importin alpha knockout and importin beta 3' UTR knockout. Initially we got the mice 8 years ago from a collaborator, Michael Bader, at the MDC Berlin. And we had the pleasure to start to work with these mice and to understand the function of importins in the neurons. And a colleague of mine, Nicholas Panayotis, started to make a screen of all the importins alpha to look at the specific of the CNS regulations, so learning and memory, anxiety, and also to look at the sensor-motor phenotype. So this was his initial investigation, in which he observed that there were a few importins that indeed had some changes in the sensory-motor function. But of course, the importins are studied also from other groups. There came up an interesting publication showing the role of importin in the neurons and the neurons function. For example, it's been published in 2018 a role of importin alpha 4, which is also called carioferin 3, in the spinocerebral ataxia of type 3, in which ataxin 3 was not going back to the nucleus in importin alpha 4 knockout mice. Another work from our lab, which was focusing on importin alpha 5 and the role in anxiety. But I don't want to go in detail talking about this interesting work, but I would like to focus on my presentation and why I started to study pain. So when I joined Mike's lab, one of the questions I had, it was if there was any importance that was important and relevant for pain, for the pain signaling. So this was the question. Is there any importance relevant for pain? And to start with this question, we thought to start with simple studies and study the acute pain. So what I did, I basically evaluated the acute pain with a heat probe, and basically I tested 58 degree with a heat probe to the paw of the mice, and we evaluated several knockout, all the knockout of importin alpha, so importin alpha 1, 3, 4, 5, 7, and basically was very exciting to see that only one importin alpha 3, had a withdrawal reflex latency delay. So these mice were feeling less pain. So that was the start of an interesting project which I follow up for these years. And the question was if these results could have been validated also with another test, if it was really true. So to do so, we decided to go and investigate the same type of similar test with the hot plate. So we decided to look at 3 different temperatures and test them. 52 degree, in which we saw that the knockout had a delay in the response, they felt less pain, and we measure in seconds. Then we also look at the 55 degree and temperature, and we saw again that the knockout mice, importin alpha 3 knockout mice, felt less pain. And the same at 58 degrees. So the mice, we could again confirm the results which we saw with the heat probe. At this point of the project, we had some idea that maybe importin alpha 3 can have something to do with acute pain. But to make sure that this was a real effect, we wanted to see with an SH RNA for importin alpha 3 if this could have been ruled out in a wild type mouse. So what we did, we created a virus, AAV9 virus, and we checked the validity of this virus by, first of all, in cell line, with N2L cells, we did an immunoblot to check the expression of importin alpha 3 with the SH alpha 3 virus, and here are the results. You see that there is a reduction of importin alpha 3. The quantification, then we checked with the overexpression of importin alpha 3, also here, and we saw that there was an overexpression of importin alpha-3. Of course, we did not only look at the cell line, we look also at the DRG because this was our goal, to look at the sensory neurons. And we started with the DRG section. So we injected the virus intrathecally, and after 4 weeks, we look at the expression level of importin alpha-3 in the sensory neurons. This is a line scan measure that we did, and we could see that importin alpha-3 was reduced compared to the wild type. At that time, we did not look also only at the DRGs, but we also look at the sensory neurons in culture, and also we look at the protein level and by real-time PCR. And we could validate that indeed these viruses were specific and were knocking down importin alpha-3. At this point, it was just a moment to start, to check in vivo if we could indeed see a reduction of acute pain by injecting the virus. So this is what we did. So we injected the virus to the wild type mice, the SH control, and the SH alpha-3. And then we measured the acute pain with the heat probe, the test I showed you before. So it was very exciting to see that with the SH alpha-3, we could see that the mice felt less pain. And we could recapitulate the effect we saw with the knockout mouse. But still at that moment, we were not completely sure, and we decided to rescue this phenotyping by injecting the overexpression of importin alpha-3 in the knockout. And please have a look at it. So basically what we did, we injected the control virus to the wild type mice, and then the control virus to the knockout mice, just to validate the data we observed previously. And we could see indeed that the mice had less acute pain. Then we injected the overexpression of importin alpha-3 in the wild type mice and in the knockout mice. And we could also see here that was rescued. So the knockout mice did not have any more the effect we saw before. It was significantly reduced. So with this specific experiment, we were more confident that importin alpha-3 had something important to do with acute pain. And of course, many of you, I think, is in the field of pain, and a major burden is also to study the chronic pain, because 25% of the population worldwide will have or is having chronic pain, and is a major concern for our society, for the socio-economic impacts. So for this reason, we decided to approach also this problem, studying the importin alpha-3 knockout mice. And how we did that, we decided to choose one model, which is the spare nerve injury model. Please, I will show you now. Have a look at the slide to the left side. So you see here is how I did it. This is the sciatic nerve, and I cut it the peroneal and tibial nerve, and I form a knot here. And I left intact the sural nerve. So it's important to leave intact the sural nerve to study the neuropathic pain over time period. We decided to study over a long time period of 3 months. We knew that the wild type mice don't recover, and we tested it over these 3 months, and the mice indeed did not recover and were feeling feeling chronic pain. But what was interesting is that the knockout mice started at 60 days to feel less pain until the 3 months. So they really recover, and they did not feel pain anymore. And what also was very striking was the paw that we took a picture at the end of the experiment at 3 months. It was a measure of a not evoke pain, in which we could see that the wild type mouse, indeed, the injury show that the paws are clenched, are closed, while in the knockout mice were open after 3 months. And this was very, very exciting, that this importin alpha-3 knockout mice had something also to do with the chronic pain. But of course, this mouse is a knockout mouse, and we wanted to see if it was also valid by a downregulation of importin alpha 3. So to so-called forming a protection by downregulating importin alpha 3 and then study the chronic pain. So let me show you what was the timeline of the experiment. So we used the AV9-SH importin alpha 3. We injected it to the mouse, as you can see in the scheme. Then after 3-4 weeks, we did the baseline to evaluate the behavior test of the mice, in this case the bone fry. And then we performed the SNI, the spare nerve injury. And then we did the assay of response to neuropathic pain check from 31 days to 90 days. So let me show you the results. So that was really striking, very close to what we observed in the knockout mouse. So in the wild type, again, we saw no differences. So there was no change. The mice was feeling neuropathic pain all over time until the 90 days. While interestingly, the SAGE alpha 3 started at 60 days, slowly improving, up to 90 days. So again, by using a knockdown viral vector of SH alpha 3, we could see that they could show that the mice felt less pain. And at the same time, we look also at an evoke test to see what also we could see from the mice after injury, if they had similar effect like the knockout. And very interestingly and strikingly, we were really surprised that most of the mice had this, in the white type was clearly that they had the clinching phenotype, while in the SH alpha 3, the toes of the paw were open. And this is a sign that the mice felt less pain. But at this point of the research, we submitted a paper, and we had a very good question from a review, and they asked us if this phenotype was specific for sensory neurons, because we could not rule out with the AV9. So that's what we did. We decided to use a specific viral vector for the sensory neurons, and we used the PHPS virus. So we did this, and first of all, we validate that indeed this virus was specific for sensory neurons. So we injected to the mice, and we checked the spinal cord and the ventral horn if there was any neurons infected, but we could not see any. So it was only the neurons infected were in the DLG neurons, as you can see here. So also by quantification, we could not detect any, and also the percentage of DLG neurons infected was about 20 to 30%. So at this point, we decided to continue and carry on and perform all the experiment again. And we use a different timeline. At this point, we wanted to understand if there was a therapeutic effect of importin alpha 3, because so far we investigated it was a protective effect because we injected the virus before and then we did the injury. In this case, we injected the virus after the injury. So let me show you the timeline. So I did SNI, that we did the baseline, that I did anti-injection at 53 days, and then what we did, we also checked the response to the neuropathic pain from 60 to 90 days. Here are the results. So basically, the SH control viruses maintain the same results. So the mice felt pain over all the time, while very interesting and striking, the SH alpha 3 PHPS virus, we could see that circa 2 weeks after we injected, we could see an effect, and the mice did not feel chronic pain anymore up to 90 days. But another good question from the review was to look at an evoked test, not only to show the paw, but also to find a way to measure if these mice felt less pain without testing them. So what we decided to do in collaboration with Nicolas, it was to put the mouse on a cage for 10 minutes and record the mouse. And then what we did, we quantified the paw licking duration. So in this case, what we saw is that the SH control had a longer time of licking the paw compared to SH alpha 3, showing that the mice felt more pain. And then when we look at the paw at the end of the experiment, 90 days, we saw clearly again that the SH control after injury had this clinching phenotype. The toes of the paw were closed, while the SH alpha 3 again were open. And this was very exciting to find out, and we want to thank the review for this very good question that allows us to think that this effect of importin alpha 3 is specific for sensory neurons. But of course, so far I describe you how is the role of importin alpha 3 in the pain, the acute, the chronic pain, and what is the phenotype of these mice. But still, we did not know the mechanism. So how this acting, how importin alpha 3 knocked mice feel less pain. And to answer this question, what we thought is that maybe importin alpha 3 is a gateway to the genome of sensory neurons and pain-related gene. So what we decided to do, we first asked the question, what about the gene expression in importin alpha 3 mutants? So what we decided to do is to look at the DRG naive and after injury. We decided to take 2 time points after injury, the SNI, so 1 week and 11 weeks. Why? Because we wanted to see exactly in the moment when we saw a phenotype in importin alpha 3 knockout mice, which were the gene differentially expressed. And to do so, we compare a 2 time point after the injury. Here you see an heat map of the RNA-seq results. So you see in the naive, first of all, you see that the genes are overexpressed are in red, 1 and 2, and downregulated are in blue, -1 and -2. So in the naive case, we see that there is a change between upregulated, downregulated gene, but then after 1 week of injury, you see a clear switch, you see a changes of gene expression. That was very clear. And then at 11 weeks, you see somehow the some genes go back to baseline. So we decided to pursue this and to investigate the knockout genome. So compare the wild type, the knockout of 1 week after and 11 weeks. And of course, we found genes which were upregulated or downregulated. But the major question was, which transcription factor is binding to importin alpha 3? So how can we study that? And we used the transpac analysis, transcription factor analysis, in which you can use your data set, your gene differentially expressed genes of the up list or the down list. We wanted to focus to the downregulated genes because of the reason that probably importin alpha 3 is transporting a transcription factor to the nucleus. So if it's not expressed, this transcription factor is not transported. So we focused on that. And you see a different groups here. And what we did a deep investigation in literature, and we decided to focus on the AP1 group. AP1 group in which there is c-Fos and Joule. We simply decided to study the AP1 group is because there are many investigations and many data in literature showing the role of FOS in pain, acute and chronic pain, is an immediate early gene. And what we wanted to do is it was to follow this line of thoughts. So why not starting with c-Fos? So what we did simply, we investigate the level of c-Fos and the localization. So let me show you this slide. So c-Fos is in the DRG neurosculpture. This is a 24-hour DRG neurosculpture plated in poly-D lysine laminin, and c-Fos is localized mainly in the nucleus, but you see also in the cytoplasm. When you compare with the knockout culture, you see the c-Fos is mainly in the cytoplasm and less in the nucleus. And when we quantified that, we saw indeed that in the knockout there was a reduction of c-Fos protein in the nucleus. And we use DAPI as well. It was a co-localization analysis. In the section also we checked the c-Fos, so we wanted to check in the DRG section, and we applied the same idea. We wanted to see the localization of c-Fos, and we saw in the white type was mainly in the nucleus, while in the knockout was localized everywhere. And this is, you see the quantification of the line bar that is in the knockout is distributed similarly, while in the white type you see the peak in the nucleus. At this point, when we saw that, we started to think that c-Fos indeed could play a role in the signaling with importin alpha 3, but we didn't know if they were interacting. So what we did an experiment, the proximity ligation assay, which allow us to investigate that in DRG neurosculpture. So we use the importin alpha 3 and the c-Fos protein. We use an antibody specific, first antibody for against c-Fos and importin alpha 3. Then we use an oligoprobe, secondary antibody against the primary antibody. And if these two oligo were in proximity, they were ligating, like you see here, and then amplification of DNA was started by DNA polymerases, and then a DNA product was formed. Then a fluorochrome oligo secondary was added to allow us to see at the confocal the puncta and the localization of c-Fos importin alpha 3 interaction. So you can see here in this example that is specific here in the nucleus. So what we did, we applied this technique directly in our DRG culture, 24 hours DRG culture, and also injury sample. So in the naive, we could see clearly the dot, the puncta of c-Fos importin alpha 3, and we quantified. While in the knockout, you saw many less. It was our control, so it was significantly less in the knockout. And then when we did the injury, SN9, after 4 hours, we also cultured the sensory neurons. We could see clearly the puncta and not in the knockout. It was significantly less. And in the white type after injury, we saw even more puncta compared to the naive. So this experiment somehow give us more confidence that c-Fos and importin alpha 3 are interacting. But there was also another way to show it, by using a c-Fos inhibitor, a drug that is already available, and there are studies in it that shows already that is important c-Fos inhibitor for arthritis. So what we did, we applied the c-Fos inhibitor drug intraperitoneal to the mice, and we had 4 groups. First of all, I don't show you here, but we did a dose response to see what was the right concentration to use, and we chose 10 milligram per kilo. And it was also published. But in this specific case, I show you data of 10 milligram per kilo of c-Fos inhibitor, and you see I have 4 groups, the white type with the vehicle, knockout with the vehicle, and white type with the c-Fos inhibitor, knockout with the c-Fos inhibitor. I test the mice after 1 hour I injected the drug, and also days post-treatment, up to 8 days. So here you see the curve, how is actually c-Fos inhibitor acting, but in this specific quantification, you can appreciate that the c-Fos inhibitor in the white type mice indeed reduce acute pain if you test with a heat probe. So we had a similar effect what we observed in the knockout mouse and also with the SAGE of importin alpha 3 virus. So when then we look at the knockout, was very interesting that indeed there was no difference. If you were injected DMSO or the c-Fos inhibitor was the same. So this was showing us the specificity and the role of the interaction between c-Fos and importin alpha 3. But of course, we did not stop there. We also look at the after chronic pain, if c-Fos inhibitor could also be analgesic. So what we did, after 1 week of SNI injury, we injected the c-Fos inhibitor at 1 hour, and then we checked at 1 hour, 5 hours, and 24 hours, a time course. And we could see that there was a clear effect at 1 hour and 5 hours, and then there was a reduction. So the effect of c-Fos inhibitor was transient in a specific time period from 1 hour to 5 hours. So at this point, I could show you that indeed c-Fos inhibitor, it was important to use to show that the c-Fos is relevant for acute and chronic pain. But of course, we wanted to go more deep and see with an SH for FOS and also for June, since it was came up the AP1 group from our RNSEC, if we could also study acute and chronic pain. So first of all, we validated the viruses. So we created an SH for FOS, and we could see that indeed in the Diogenes culture, when we stained for FOS, there was a reduction. With 2 types of SH for FOS, there was a clear reduction, and also for June. At this point, we said, okay, we want to continue, and we're going to inject the virus to the white-haired mice. So we wanted to study, as I said, again, the acute pain with the heat probe and with the Wolfram. So with the heat probe, we could see that indeed the downregulation of FOS was sufficient for reducing of acute pain, measured by with the heat probe, and also with the SH June. Then we also look at the Wolfram, and we didn't see clear effect with the c-Fos, but with the c-Junes, we saw a change in the response to the air filament. Of course, our goal was to study the chronic pain. And to do this experiment, we decided to include also the CH-alpha-3, because to repeat our results, if they were also possible to study after the injury. And we use the AV9. So in this case, we use an AV9 and we inject the virus not before injury, but after injury. So we injected, so this was AV9 injection, and it was after the injury to show the therapeutic effect of the virus. And then we studied all these viruses in parallel, and we could see that indeed, just to show you, the CH-alpha-3 in red, it repeated the data we observed before. So the mice felt less neuropathic pain, and the CH-June here in violet, also the mice felt less neuropathic pain, and the same also SH-Fos. This was studied over 3 months time period. And this was very exciting. And I would like to actually sum up what I told you so far and give you the most important results we observed. So first of all, Importin alpha-3 and c-Fos interact, controlling the expression of pain-related genes. And how does it happen? First of all, we saw that the loss of importin alpha-3 just by reduction with an SH or with a global knockout reduces acute and chronic pain. The loss of importin alpha-3 also reduces the nuclear import of the transcription factor c-Fos. And c-Fos and importin alpha-3, they interact. Another important result was the downregulation of c-Fos by drug or by an SH RNA with the virus, or also the downregulation of June, the AP1 group, reduces acute and chronic pain. But there was still one question left, and I want to tell you. Can we target this mechanism for pain therapy? How can we study that? We know that importin alpha 3 and c-Fos are relevant for the acute and chronic pain. So how can we study which are the drugs that are available and maybe apply those drugs to study neuropathic pain? So I don't know if you know, but there is this cognitive map analysis from the Broad Institute that we used. It's a very good platform. CMAP is a genome scale library of cellular signature that catalogs transcription response from chemical, genetical, and disease perturbation. CMAP has generated a library containing 1.5 million gene expression profile from 5,000 molecular compounds tested in multiple cell line of human cell line. So what we did, we use our list of genes, different genes expressed in importin alpha 3, and compare with the data set that was available in the CMAP analysis. And what we obtain a signature of candidate drugs that are very similar to your data set, to the importin alpha 3 knockout in our case. So you get a CMAP score that goes from 1 to less to 0. And of course, we focus on the first, at the closest to 1, because that drugs were very similar to our signature of c-Fos importin alpha 3 signal. And then, of course, we checked for side effect of the drug, what was known, if it was FDA approved, if there was general cellular modifier, and if it was already studied in pain. So we look at all this carefully in literature and find out that there were 3 drugs that could have been tested for pain. That was ashmalin, that is an antiarrhythmic, sulfametazole is an antibiotic, and sumazole is also an antiarrhythmic and cardiotonic. So basically, I show you what was the experiment. Here is a chemical structure of the sumazole and sulfametazole that indeed they are very similar between. So that's why I wanted to show you this. But let's go to the data. So basically, we injected the drug intraperitoneal, and after 1 hour, I tested with a heat probe. And I saw that the sumazole and sulfametazole indeed had an effect on acute pain, and they were the most interesting drug to me. At this point, we decided we might to investigate also the chronic pain. So what we did, we injected the drug at 2.5 months after the SNI injury to white-type mice. And we inject also the vehicle. And what we did, we evaluate after 1 hour of injection of the drug, the effect in chronic pain. So this was also very exciting, that this drug was able to reduce chronic pain in the white-type mice. But there was another question of the review, very good one, to ask, is this effect also applicable in a shorter time period, not only at the longer time? So we studied the drug at 7 days after SNI. So we studied sumazole and sulfametazole, and we did a time course, and we checked at 1 hour, 5 hours, and 24 hours. At the end, we saw that only at 1 hour we saw a clear effect of sumazole at 7 days after SNI. So these results so far show you that these 2 drugs specific, they are mimicking the effect of importin alpha 3 c-Fos signaling. But there was still one question lacking from the review, was to study the acute mechanical pain in the injured leg. How do you do that? You have your mouse that was already 3 months after the SNI injury, and we wanted to test the 2 gram filament, so to a heavy acute mechanical pain to already an injured leg. And to do so, we decided to use a response score that it is considering 1 like a light discomfort, so means the mouse is not moving the leg but is feeling discomfort, and 2 is when the mouse is moving the leg after poking with the 2-gram filament. And this is we applied in our model, wild type and knockout mice, and also with the SAGE control and the SAGE alpha 3. Plus, we wanted to study the drug effect 1 hour after the injection of the drug. So this was really striking. If you see, basically, the wild type mice and the SAGE control felt pain clearly. So they moved the legs after poking with the 2 grams, while the knockout and the SAGE alpha 3 did not feel the same pain. And the same, very interesting, also the sumazole, sulfametazole drug was feeling less pain. And this is was a common feature of all these 3 type of inducing drug, and also a SAGE, or even knocking down importin alpha 3. So the question was if sumazole and sulfametazole have something in common with c-Fos. So there is a reduction of c-Fos, for example, in the nucleus. And what we did, we investigated in the DLG neurons culture, 24 hours culture, the vehicle and the sumazole in the wild type culture and in the knockout culture. Here the image you see the wild type culture. So what you see is that in the vehicle stained with Juan, CGRP and c-Fos and DAPI, you see that c-Fos is clearly in the nucleus, and it was quantified here, the c-Fos staining, if it was more in the nucleus or more in the cytoplasm. And you can clearly see that in vehicles is more in the nucleus. Well, when you add drugs like sumazole or sulfametazole, you see that the c-Fos is avoiding from the nucleus. And here is the quantification. You see a clear reduction of c-Fos from the nucleus. And the same results we obtain with the knockout. In the knockout, if you add the vehicle, sumazole or sulfametazole, you always saw a reduction of c-Fos in the nucleus. So with this, I would like to summarize and tell you that what we think, that we find a novel way to investigate acute and chronic pain with importin alpha 3, retrograde transport, and c-Fos. And if you reduce c-Fos by downregulation, there is a reduction of acute and chronic pain. If you reduce importin alpha 3 by reduction or globally, you see a reduction of importin alpha 3, and you see also that in the knockout of importin alpha 3, c-Fos is reduced in the nucleus. Plus, we could show you that pharmacologically, with sumazole and sulfametazole, 2 drugs FDA approved that were not before known in pain, are now possible to study, and future investigation will be done for this type of research, for therapeutics. So I also would like to thank all the people that were in these studies. A big thank to Mike Feinzibar, that first of all he chose me when I came for the interview, and he gave me 2 very interesting projects. This one I present to you is the side project, and there is another project I'm working on and finishing up. And it was really beautiful to work with Mike, and I want to say a big thank to all the colleagues that are great, and without them, of course, these great results could not have happened. And a special thank to the people that were closer to me and to the project. To Nicolas Panayotis here, you see him, Stephanie Alba here, that she left the lab, and then Natalia here, and Indrek Koppel here, and of course all of them. I also want to thank the collaborators, Mikael Bader Lab and Franziska Rother. Also would like to thank Enno Hartmann, our collaborator, and I don't want to forget all the professional support I got over these 6 years from fantastic people, scientists that could help me in any type of research. So with this, I also want to thank you that you could listen to me over this time. And this was my first seminar, so I'm very glad I could talk to you a bit about my project of my last 6 years work. So open questions, and I'm available to discussion. Thank you.
Thank you very much. That was really terrific. So we have a number of questions. I wrote down several. I know other people have questions too. And I think the best way to do this is for y'all to raise your hand so that there's a little raise hand button. And I see that Zach Campbell has his hand raised already. So Zach, I'll let you take the floor and ask the first question.
Thanks, Ted. Really beautiful talk, Letizia. Appreciate you doing it so much. So I just had, well, I have a bunch of questions, but I'll boil it down to the one that is the most curious. What do you think the relevant targets of c-Fos are in the context of sensitization? Because, you know, it's pretty pleiotropic. So I just wonder if you thought downstream about what might be underlying the regulation. Thanks.
And can you repeat again? I could not hear.
Sure. So what do you think the relevant targets of c-Fos are in the context of sensitization?
c-Fos. Okay. The gene. So you're talking about the gene of c-Fos. The
transcriptional targets that are downstream of c-Fos.
Of course. Yes. So this is a great question, and this is we haven't investigated yet deeply, and it's something that we would like to do, and to look at the genome and look at the target gene of c-Fos, to be able to see if we look at this genus, we knock it down, we see the same effect. And this is we haven't started yet, but it's something that we have in mind. Yes.
All right, we got another question. This one in the chat box from Saad Yusuf, who's a postdoc in my lab. So he's asking, have you started to look at any potential sex differences in the alpha-3 Importin knockouts?
This is also a very good question. So the neuropathic pain analysis we did were in the male mice, and this is what we would like to start now, to look at the female mice, if we can see the same effect in the Importin alpha-3 knockout mouse as a start, and then with the viruses, and also if it's the same signaling of c-Fos. So this is a very important question, and we are starting now to look at.
Mm-hmm. All right, so I'm going to ask a question now. So, and I think some other people had the same question. So in the pain field, in neuropathic pain in particular, a lot of folks are usually looking at like the 7-day, 14-day time point. Some people go out to 21 or 30 days even, but rarely to 60 days. So your data is extremely persuasive that something really important is happening at that relatively late time point that a lot of people don't study. And I guess my question is not so much a science question or like a mechanistic question as it is just, how did you guys decide to stick with it and keep looking at the phenotype of these mice for so long? What were you thinking when you had a clear acute heat pain phenotype, but nothing was happening in the SNI model? And I'm just kind of curious about the thought process behind the experiments, and if you guys were particularly struck that something happened at such a late time point, or if there was some underlying hypothesis that led you guys to look at that.
This is also a very good question. So we initially wanted to go to 2 months because there was one publication showing that, they start investigating neuropathic pain, the white-eyed mouse, and we drag up to 2 months. So this was our target time. And then when we initially start to see some difference 60 days after SNI, we could not have stopped there because it was really initial. And it was not clear if it was maybe a mistake due to the day or it was a real evident effect. So I think the decision was made after we got to the 2 months. Until then, we did not have a clear idea that Importin alpha-3 has a role in chronic pain. We knew about the acute, but we was not sure. It was not something we knew it from other data. I hope I could answer.
Yeah, that's it. Yeah. So another question related to that is, your lab has shown previously that there's local translation of a beta subunit of important that then it undergoes retrograde transport. Do you guys think that there's a similar potential mechanism that's driving local translation of the alpha subunits of importance, and that they're transported back, and that there may be differences in early versus late alpha subunits that are locally translated?
It's a good question. We haven't looked at it, so
I see Mike's hand went up too. So
what?
I see Mike's hand went up, so I think he wants to chime in on this particular one.
Okay, go ahead. If he wants to answer, no problem. But at the moment, for the data we obtain, we haven't looked at this specifically of the locus translation of Importin alpha 3 index. So this is what I can say from my data.
Mm-hmm. Mike, did you want to chime in? We can't hear you though, unfortunately.
No, I cannot hear.
We can hear something now.
Okay,
Yes.
is this working now?
Yeah, we can hear you.
Okay, so Jeff Twist has some, in some of his data sets, we do see some of the important alphas that may be locally translated. But we have not really looked in any great detail at regulation at that level. Mm-hmm. And actually, one of the puzzling aspects of Letizia's work and also Nicola's work is that when we were looking at all of these mutants of Importin alpha, we were expecting axonal presynaptic phenotypes, and what we actually see are mainly nuclear import phenotypes. Sorry, we were expecting postsynaptic phenotypes. So we see nuclear import phenotypes. Mm-hmm. We don't see actually Importin alpha regulation as something which is happening, at least in these models, pointing to things happening in the axon, which either suggests that these complete knockouts are not sophisticated enough to show what is actually happening, or that it's not happening. Whereas the Importin beta 1, we do know that there's local translation regulation on multiple levels.
Yeah.
Anyway, I'm now going to mute myself and stop stealing the stage from Letizia.
No, you're welcome.
All right, I got a question here in our chat box from Aish, who's a PhD student in my lab. So she asked, does Importin also bind to other transport proteins that are required for regulation? For instance, ARL8. Also, does the level of Importin change after injury?
Importans are binding transcription factors. This we know exactly which Importin alpha is binding which transcription factor. There are some studies in the lab that will elucidate this, but also with BioID. But in this specific protein, I did not find that was binding Importin alpha 3. But I haven't checked this, so I was mainly focusing on c-Fos.
So the second part of her question is, do the protein levels of Importin alpha 3 or other importance change after injury?
I checked that, and I haven't seen a clear effect. Just not significant. In the time course, we look at 1 week and 11 weeks after injury. So that there was not a clear change at the gene level.
So something that I think is quite striking in the data, and we often hear this kind of complaint from reviewers about our own data, is that you're manipulating a transcriptional pathway. In our case, we're usually manipulating a translation pathway, and we see very rapid onset behavioral effects. So in your case, within an hour. In our case, usually 15, 20 minutes. And I think that falls a little bit outside of the expectations of most neuroscientists in terms of gene expression changes leading to behavioral alterations. So I'm curious if you guys could just, if you could comment just a little bit about the.
I can comment. So I also was curious about this because FOS is an immediate early gene, and it was kind of predictable that has an effect in acute pain, but less predictable that have an effect in the chronic pain. So what I did, I went to check the level of FOS and AP1 group in our gene data set at 1 week after injury and 11 weeks. And what I could see that there was an upregulation in the Y type of FOS gene. And this was not studied before because, as you also mentioned before, the studies of neuropathic pain is going up to 1 or 2 months. And this RNA-seq was done 11 weeks after injury. I don't know if I could somehow answer your question.
Well, so I mean, I guess the question is more about like the dynamics of how rapidly these transcription factors are able to change behavior. So like when you give the sorry, I can't remember how to pronounce the drug, but you had two that started with an S. That manipulate c-Fos signaling, and then you give them and they have an effect within about an hour.
An hour.
Yes. So I mean, have you guys done any experiments to try to look at just how rapidly these drugs have an effect on transcription, and or to try to understand? Again, I guess this goes back to a previous question. How these quick changes in transcription might lead to changes in behavior.
And this we did not check. So this is also a good question, and also to look at the gene level of this specific drug, c-Fos inhibitor, look at 1 hour after the gene level in the Y type and the DLG. We haven't checked that. This is actually a very good question. Can be pursued in future.
Yeah, I guess this is something that all of us in the gene regulation and behavior fields are trying to kind of tackle, because it seems like these effects sometimes occur on a more rapid time scale than most people expect that they would, based on what we know about transcriptional and translation regulation. Or I guess I shouldn't say what we know, what we think that we know. So, along those lines, and again, I don't want to steal the discussion, but, Aaron Schumann's lab just had a paper deposited on bioRxiv where they were using reporters looking at translation in dendrites in LTP and LTD. And, they were showing bursts of translation that happen within seconds in dendritic spines, which kind of just blows my mind. But, the data is extremely persuasive. So, okay.
Yes.
I think Saad had his hand up, but then his hand went down. Saad, did you have another question?
Yeah, I actually had a couple,
Great.
but
Okay, great. Go for it.
Well, I was wondering, do you think that there is another factor that may be important? So, like you mentioned that importin alpha 3 does not go up with injury. Would you expect there to be another factor, for example, one of the beta importance that is responsible for that transport across the nucleus, or maybe the nuclear transporters themselves? Has that something that you've been looked at?
But importin alpha 3 is expressed in the sensory neurons as a protein, And the difference between importin beta and importin alpha is that importin beta is mRNA and then is locally translated upon injury, while importin alpha is already there in the axon as a protein. So the question if maybe there are transcription factor, specific transcription factor beside also c-Fos that might regulate the signaling on pain. There could be. But importin alpha is making a heterodimer with importin beta. So and the importin alpha is binding the NLS sequence of a cargo, a specific cargo, which in our case we investigate c-Fos. So the question about the local translation of importin alpha 3, it might be importin beta locally. Importin beta we know is locally translated upon injury, but we haven't checked a long time period after. We checked early after injury in the sciatic nerve. So this could be a good question to check later on, like in the time frame I check neuropathic pain, if importin beta is locally translated, for example, 2.5 months after SNI. This is something we haven't checked, but it could be also a good question, a good studies to do. If Mike has other ideas.
His hand popped up again, so go
Yeah,
ahead.
I just wanted to mention there's a paper in bioarchives where we have a small role from Jeff Twist's lab. It's Sahu et al. Currently being churned in current biology after a massive revision process. And Kabi Sahu there looks at the regulation of local translation of importin beta just as one of the comparisons. So you can see there's some aspects of the kinetics of translation and the dependence of translation on calcium that may or may not be relevant to these types of models that we, more relevantly, you guys are looking at. So I think one of the problems with looking at regulation of importin beta 1 is that it's critical for a lot of nuclear transport. So I mean, about 2/3 of nuclear import is dependent on importin beta 1. So it's a very major thing to change if you want to look at something regulatory. There are a lot of other importin beta family members that have not been looked at in detail. We looked at transportin for a while, which is not translated, but is present in neurons and has roles. And there are other family members that probably have roles. So if any of you guys want to look at that, please. We're very interested to learn about it.
Yeah, I think we're pretty interested. Saad, go ahead. Yeah.
Can I just I just had another question. So as far as I remember, c-Fos is still translated. c-Fos is still expressed early on in that acute phase, right?
Yes.
But there's something that happens at that day 60 time point, which allows importin alpha 3 to be able to translocate it into the nucleus. Is that correct?
So what I see from the gene, so from the RNA-seq, I see that importin alpha in the importin alpha 3 knockout mice when I compare with the wild type, and I see different time point. I see that FOS is upregulated after 11 weeks after SNI. So at 7 days you don't see so high, and then 11 weeks goes really up. And this is what we have. We haven't studied at the protein level in the DRGs at this time point. We always studied 4 hours after SNI. All the data show you in the culture it was always a short time period when you see the peak of the protein in the nucleus going up. But
okay. Sorry to cut you off, but I just wanted to confirm that c-Fos doesn't really go up acutely, but only goes up in expression chronically at that 11-week time point. Is that right? Okay,
yes.
Perfect. Thank you for clearing that up.
For sure.
All right, I don't see any more questions in the chat box, and nobody's hand is up, so I'm going to take the initiative to ask the last question. So you very kindly spent a lot of time answering our questions, so thank you for that. So my last question is just on a methodological thing with the S&I. So, and it's not so much a question as it is commending you on looking at this paw stature, I guess we could say, because
Yes.
I think that this is something that a lot of people in the neuropathic pain field have noted over and over again, but few of us have quantified. And I think it potentially is a really nice measure of spontaneous pain. I mean, how else do you really explain it? I guess there could be some motor things that are involved, but I think potentially it's a really nice measure of non-evoked pain, and people in my lab, I think, know that I feel pretty strongly about that, because I've been talking about this for a long time, that we could potentially measure it. So I guess my question is just, how robust has this been as a measure for you guys? And are you just taking pictures and
Yes.
scoring? Yeah, is that how you're doing it? Yeah.
So basically, before sacrificing the mice, so as I said, it was 3 months, the time period that we evaluate the neuropathic pain. So first thing, before sacrificing the mice, we take a photo of each mouse. So we have a PowerPoint presentation for each mouse to look at the not injured leg and injured leg. So we have a map of all the mice we injured, and we can see when there is an effect. More than 50% of the mice, 60-70% of the mice at this opening. But ours is only observation, because our photo we haven't quantified, maybe as you suggesting, with some software to look at the area, I don't know, of the paw. But I think it would be a good idea to add the photo some more quantification and give a value, statistical value of the trend of the mice in the experiment. I think it's a good point that at that time we did not think to do it, because actually it was the first time I saw myself, and I haven't seen so much in papers doing it. So said we can try to show it, but.
I've never understood why people don't quantify this more often. Mike, go ahead.
Good. How would you think to quantify it? I mean, would you suggest that one would measure the distance between the toes or something like that? What are your thoughts?
The area.
So here's the thing that we have been kind of thinking about, but although we haven't really done it yet, and I'd be curious if you guys have seen this too. So the paw position is a little tough because it can change, depending on where the mouse is, etc. We've noted for many years now that if you pick a mouse up by the tail, right, that usually they splay their feet out like this, their hind feet, right? So my head is the tail, and then their feet go out like this. But neuropathic mice always do this.
Mm-hmm.
But so did you guys also note that they did this up until the 60-day time point, and then they started like this again? Or did you ever even?
I noticed the mice were really not like the leg and the position was more normal, and but I did not quantify. I haven't done a very precise like I did for the paw. So when I was holding them, of course, to put in the setup and any type of manipulation, I could see that they were different in the position, the legs.
There's a mouse called the LOA mouse. I guess that may be familiar, from Elizabeth Fisher. It's one of the original dynein heavy chain mutants, and LOA stands for legs at odd angles.
I'm actually not familiar with this at all. No, I had no idea.
It's a massive science paper from a decade and a half ago, Hafez Parast et al. And what they actually did, it's a point mutation in dynein heavy chain 1. It came from an ENU mutagenesis screen in Elizabeth Fisher's lab and University College London. And this is a sign of neurodegeneration. You pick the animals up by the tail. This is how you genotype them, so to speak. You pick them up by the tail, and if they have this clenched legs phenotype, then you know that those are LOA mice. So the fact that you say that it's similar in neuropathic pain is actually striking for me.
Yeah.
I mean, I will send you the reference. It's
Yeah,
well,
please
it's a very famous paper for Dainian aficionados.
Okay. I'll take a look, and yeah, if you remember, please do send it to me. I'd like to see that. Yeah. So we, it would be interesting to maybe apply some machine learning type ideas to looking at some of these images and just see what we could maybe do collectively to create a score for this, because I do think it would have a lot of translational potential.
Yes.
And having non-evoked pain measures is really important. As you know, the field is all looking at this, and I've just kind of been stunned that nobody has taken more advantage of this. But we're our lab, who creates as many neuropathic pain mice as any lab in the world, probably. We also haven't done it. So kudos to you guys for doing it.
Okay. Yes.
Yeah.
Good point.
All right. Well, Letizia, thank you so much for a really, really great presentation and really great discussion. Mike, thank you for joining us, and I'm sure that we'll be in touch with you guys about this last idea we talked about, but also lots of other ideas related to your really great work. And congratulations on your soon-to-be released paper.
Thank you, Ted. Thank you for the invitation.
Yeah, my pleasure. Okay. Thank you. Bye, everybody.
Bye.